All right, let's jump right into this explainer. Today, we're going to explore the absolute marvel of biological engineering that is your eye's blood supply. It's a place where structure completely and utterly determines function. Even though the eye only accounts for a tiny, tiny fraction of your body's total mass, it actually possesses one of the richest, most highly regulated vascular systems in the entire human body. Every single second, your retinal neurons are just devouring enormous amounts of oxygen. And even a brief interruption in that pipeline, well, that can mean permanent vision loss. So here's our roadmap for today. We've got 5 main sections: the eye's blood supply, the dual circulation system, when blood flow fails, visualizing retinal blood flow, and finally, we'll wrap up with our clinical takeaways. Starting with section 1, the eye's blood supply, tracing the path of perfusion. Okay, so every single tissue in your eye gets its arterial blood from exactly one source, the ophthalmic artery. We can literally trace a drop of blood on its journey. It emerges from the cavernous sinus into the internal carotid artery, and from there, it flows straight into the ophthalmic artery, which actually enters the orbit right alongside the optic nerve. And that intimate anatomical relationship is exactly why vascular and neuro-ophthalmic diseases are so often linked together. Once it's inside, it branches into the central retinal artery and the posterior ciliary arteries. Now, the central retinal artery is absolutely crucial because it's essentially an end artery. It pierces the optic nerve, spreads across the inner surface of the retina, and supplies all your ganglion and bipolar cells. Now, this is wild. Despite the retina getting pretty much all the spotlight in clinical discussions, a massive 80 to 85% of all ocular blood flow is actually directed to the choroid. The choroid is supplied by those posterior ciliary arteries we just talked about. It is this intensely vascular layer with highly fenestrated, meaning leaky, capillaries. And this massive volume of blood is non-negotiable because the outer retina, specifically those photoreceptors, are among the most metabolically hungry tissues in your entire body. They're constantly regenerating, constantly recycling, and they need an astonishing amount of oxygen just to keep you seeing clearly. Moving on to section 2, the dual circulation system, an elegant evolutionary solution. So this brings up one of the most brilliant concepts in ocular physiology. The retina actually relies on 2 completely separate circulatory systems. But you might be wondering, why? Why did evolution develop 2 independent vascular systems for the eye instead of just building one unified network? It kind of seems like an unnecessary complication, right? Well, the answer comes down to competing physiological demands. See, the inner retina absolutely must remain transparent. If you had giant, massive blood vessels extending deep down into the photoreceptor layer, they'd scatter the incoming light and totally ruin your visual acuity. So the inner retina gets a very delicate, transparent supply from the central retinal artery. But those outer photoreceptors, like we said, they are incredibly hungry for oxygen. They literally can't survive on that delicate inner supply. So they rely on that massive fenestrated blood flow from the choroid, which safely sits beneath the retinal pigment epithelium, delivering oxygen by diffusion without ever blocking the light. Honestly, it's the perfect compromise. Now, to protect this super delicate inner system from the wild, everyday swings of your systemic blood pressure, the retinal vessels use something called autoregulation. And this is fascinating. Unlike most other blood vessels in your body, once these enter the eye, they aren't controlled by your autonomic nervous system anymore. Instead, they dynamically dilate and constrict based purely on local metabolic factors like tissue oxygen levels and pH. So if your blood pressure spikes, the retinal arterioles constrict. If it drops, they dilate. It's an ingenious system that ensures oxygen delivery perfectly matches the continuous demands of your retinal neurons. Section 3. When blood flow fails, the anatomy of an ischemic emergency. But what actually happens when that autoregulation fails or a total blockage occurs? Let's break down the catastrophic anatomy of an ophthalmic emergency. Remember, retinal neurons have practically zero anaerobic reserves. They just can't survive without oxygen. When blood flow stops, an ischemic cascade triggers in literally seconds. Oxygen drops, shutting down oxidative phosphorylation. Without that, your ATP energy production just flatlines. And without ATP, the cellular ion pumps fail completely. Sodium rushes into the cells, dragging water right along with it, causing the cells to visibly swell up. Finally, toxic amounts of excitatory neurotransmitters like glutamate build up, causing excitotoxicity and ultimately irreversible infarction. This whole cascade leads us to a classic clinical presentation. During a central retinal artery occlusion, CRAO, that inner retina rapidly suffocates. It swells up and turns a very pale, opaque white. But this creates a really fascinating micro-mystery. If the entire retina is starving and turning white, why does the central fovea stay a bright, vibrant red? The answer is incredibly elegant, and it brings us right back to our dual circulation. The fovea is anatomically unique because it's extremely thin, and because it's so thin, it actually doesn't even need the central retinal artery. It survives entirely on oxygen that diffuses directly from the massive, still-functioning choroidal blood supply right beneath it. So while the surrounding inner retina basically dies, swells, and turns pale, that underlying red choroidal blood remains completely visible through the surviving transparent fovea. And that right there is the famous cherry red spot. So the real crucial takeaway here is that the specific vessel that gets blocked perfectly dictates what you see in the clinic. If it's a central retinal artery occlusion, it's sudden, it's a total inner retinal block, and you get that cherry red spot with sudden painless vision loss. Now, if it's a branch retinal artery occlusion, well, only one arteriole is blocked, so you just get a sectoral kind of pizza slice-shaped area of whitening. But if the problem is higher up the chain, say a blockage in the carotid artery, you get ocular ischemic syndrome. That's gradual, the whole eye is basically hypoperfused, leading to dilated veins and persistent chronic hypoxia. And let me tell you, chronic hypoxia is a massive problem. When retinal tissues are starved for oxygen over a long period, they literally panic. They start pumping out a signaling molecule called VEGF, vascular endothelial growth factor. VEGF basically screams at the eye to grow brand new blood vessels to try and get more oxygen. But it's a completely flawed rescue attempt. These new vessels are incredibly fragile. They lack proper tight junctions, so they just leak fluid and bleed everywhere, leading to devastating downstream complications: vitreous hemorrhage, fibrous scar tissue proliferation, and eventually tractional retinal detachments that can permanently destroy whatever vision is left. Section 4. Visualizing retinal blood flow, fluorescein angiography in real time. To combat these emergencies and figure out exactly what's failing, modern medicine relies on a truly ingenious tool called fluorescein angiography. Basically, we inject a fluorescent dye into a patient's arm vein and use a special camera to watch it hit the eye in real time. We can literally map the dye's dynamic passage. At about 10 seconds, boom, we see a diffuse background glow. That's the choroidal flush, filling fast because of those leaky fenestrated capillaries. Then, just 2 seconds later, at 12 seconds, the dye sweeps strictly into the retinal arteries, pushes through the whole capillary network by 15 seconds, and finally drains out via the retinal veins right around 20 seconds. It happens so incredibly fast. By watching this flow, we aren't just looking at a static anatomy picture. We are dynamically stress testing the plumbing. If the vessels are sharply defined and the retina around them is dark, awesome. The blood retinal carrier is healthy. The tight junctions are working. But if we see these dark, non-fluorescent gaps where capillaries should be, we know immediately we're looking at dead, infarcted tissue, capillary non-perfusion. And if we see blurred vessel margins that get intensely bright and glowing as the test goes on, that's leakage. That tells us those fragile VEG-driven new vessels are there, and they're actively leaking dye right into the retina. Oh, and here is a surprising and just beautiful fact to remember. On a completely healthy angiogram, the central fovea shows up completely black. It is entirely avascular. And this perfectly proves our earlier point. There are absolutely no retinal capillaries in the very center of your vision. It is kept perfectly clear to prevent any blood vessels from blocking the light hitting your most important cone photoreceptors. Nature's design at its finest. Finally, Section 5, Clinical Takeaways. Structure determines function. Okay, let's pull all of this beautiful biology together. When a patient sits in your chair complaining of vision loss, all of these biological mechanisms empower you to ask one simple but incredibly powerful question: where is the problem? If the clinical signs point to the retinal arteries, like you see that pale inner retina, you're immediately thinking emboli or thrombosis. If the posterior ciliary arteries are the ones failing, your brain should jump to optic nerve head ischemia or giant cell arteritis. And if the veins are dilated and actively leaking, you're looking at venous hypertension and edema. The structure of the eye entirely dictates your diagnostic path. So ultimately, the key takeaways from today's explainer are pretty clear. The eye's dual circulation really is a masterpiece of engineering. You've got the central retinal artery carefully supplying the inner retina, while the posterior ciliary arteries just flood the outer retina and choroid. Remember, retinal neurons simply cannot tolerate oxygen loss. That ischemic cascade is brutal, and it is fast. While autoregulation tries its absolute best to protect the eye, chronic hypoxia inevitably leads to VEGF release and those destructive new leaky vessels. And through it all, dynamically visualizing this flow with tools like fluorescein angiography remains our greatest weapon in diagnosing these sight-threatening emergencies. Which leaves us with a genuinely thrilling question for the future. We've seen just how catastrophic it is when retinal blood flow fails. But as our imaging technologies continue to advance, we're talking about things like non-invasive OCT angiography, precise blood flow quantification, and new vascular biomarkers. How much earlier will we be able to intercept this ischemic cascade before the irreversible damage ever even begins? Thanks so much for joining me on this explainer. Keep wondering, keep learning, and definitely think about that question until next time.